Hello everyone, welcome to eDufundae. I am your mentor for the topic Reproduction in Bacteria. So let us understand the reproduction. The bacteria reproduce commonly by a kind of cell division, that is binary fission. The dividing bacterial cell produce two genetically identical daughter cells. The interval of time between successive binary fission of a cell is known as the generation time or doubling time, which may be as little as 20 minutes in some bacteria. So basically जब हम बैक्टीरिया में रिप्रोडक्शन की बात कर रहे हैं एंड पर्टिकुलरली हम एसेक्सुअल रिप्रोडक्शन की बात कर रहे हैं तो उसमें हम नोटिस करते हैं दैट बाइनरी फिजन इज टेकिंग प्लेस. अर्लियर क्लासेस में हमने बाइनरी फिजन अमीबा के केस में पढ़ी है दैट अमीबा इज डिवाइडिंग इट्स ओन बॉडी इनटू टू एंड द टू डॉटर cells are formed. Similarly, over here the two bacteria will be formed after binary fission, and this doubling time or the generation time can be as little as 20 minutes in some bacteria. And if I talk about sexual reproduction, the true sexual reproduction is absent in bacteria. The exchange of genetic material, that is genetic recombination, which is the essence of sexual reproduction, takes place in three ways. The first one is sexual reproduction by conjugation, transformation as well as transduction. So these are the three ways of the sexual reproduction in case of bacteria, which is not actually the true sexual reproduction. It is only the exchange of genetic material, that is through conjugation, transformation and transduction. So let us understand first conjugation. In this process, two live bacteria come together and the donor cell directly transfer DNA to the recipient cell. That means if there are two bacteria, one will be the donor and the other will be the recipient. And what is the donation of taking place? Of DNA. This process was first observed in 1946 by Lederberg and Tatum in Escherichia coli, that we also call as the E. coli. Let us see the representation of the process of conjugation. Here we can see two bacteria. One is the donor bacterium and the other is the recipient. The recipient is having a blue colored ring-like structure and it is the F plasmid. Why F? Because it is the fertility factor. Now this donor will be donating the genetic material to the recipient with the help of a bridge-like structure that is called as pilus, and once the donation is done, the recipient becomes the new donor. So here we see that here is the F plus plasmid, or also called as the F1 plasmid. It is the fertility factor, but in recipient there is no F factor and that is why it is F negative. Now there is this bridge-like structure that is pilus. The plural form is pillae, and here we can see that there will be the formation of the pillae in both the structures, that is the old donor was already having it and the new donor can further donate the genetic material, so it also has this pilus. The process of conjugation requires a special conjugation apparatus called the conjugation tube or pilus or the sex pilus. They are all same. So here we see that there is the presence of two bacteria and the formation of pilus is taking place in between, and the purple colored plasmid is moving towards the recipient, and this is how the conjugation is taking place. For cell-to-cell contact, the donor cell designated F plus produces the pilus that makes contact with the recipient cell known as F negative cell. The donor cell is called F positive because it contains a plasmid called as an F plasmid. The F negative cell lacks a F plasmid. Once contact is established, the pilus shortens to bring the two bacteria close together. The F plasmid then begins replicating and replicated DNA passes through the bridge formed by the pilus to the recipient cell. in that donor bacterium generally retains a copy of genetic material being transferred. So we can say that conjugation is a very conservative process. Next is transformation. This mode of bacterial genetic recombination was discovered by Frederick Griffith in 1928 in Streptococcus pneumoniae. Transformation is uptake of naked DNA fragments from the surrounding environment and the expression of that genetic information in the recipient cell. That is, the recipient cell has now acquired a characteristic that is previously lacked. So what we see in this process, that the naked DNA fragments are directly being uptaken from the surrounding environment by the recipient, and the recipient cell has now acquired a characteristic that was once not there in its body, but now after acquiring it from the environment, it is having it. This discovery of DNA as the genetic material was based on transformation. So here we can see the process of transformation. Once the bacterial chromosome is there in the environment of the other bacterial cell, it is simply taking it up and it will inculcate it with its own plasmid and becomes a transformed bacterium. So transformation process. The next process is that of transduction. In this, the transfer of genetic material from one bacterium to another is through bacteriophage, is known as transduction. So basically what is happening in this particular process, there is no pilus, there is no other environmental genetic material. Basically the virus that is called as bacteriophage is the virus that infects and replicates within the body of bacteria, and with the help of it, the exchange of genetic material is taking place. So here we see in this representation that there is a bacterium in which there is the presence of a plasmid. Now the bacteriophage, that means the viruses, are attacking the body of this particular bacterium and they will inculcate the genetic material taken from the donor to this recipient bacterium. It was discovered in 1951 by Lederberg and Zinder in Salmonella typhimurium. Let us see the representation of transduction. Here we can see there is one donor bacterium to which the bacteriophage is attaching. Now the bacteriophage is entering inside the body as well as replicating inside. Now it will take up the A plus and B plus, or you can say A positive and B positive kind of plasmid from the donor bacterium. Now what its function is, it is carrying the phages, or you can say the donor genes. They are the donor genes that will be further entering the body of the recipient bacterium. Now recipient bacterium is having the opposites of what it carried from the donor bacterium, and the same location from which it came from the donor plasmid, they attach in the recipient plasmid, and it leads to the formation of the transducted bacterium. So here you can see in the last picture towards the left, there is a transducted bacterium that is having the A positive fragment of the plasmid that came from the donor bacterium. So this is how the process of transduction takes place. I hope you understood the explanation. Thanks for watching.